Much to do today, as it were. So we are recording, and you might recall that we left off. I'll just show you briefly. We kind of left off here. We had finished, and I'm not going to spend any more time. If you remember the last ones, that all the different energy molecules and proteins, because that's all AP1, and there's no need to rebuild the wheel, so to speak. So we're going to talk. We're going to start here, but I want to present this stuff first. You're going to find a lot of it that I'm going to be reiterating it's on Blackboard from the additional notes, chapter 1 micro with that 2 in parentheses. That's really what we're talking about today, more than anything else. And so if you look on that board, and pardon my bad handwriting, which is why I type as opposed to writing, because my handwriting is atrocious. Where did I go? So when something is living, by definition, it's made out of cells. Most importantly, it's got both DNA and RNA. For living things, to our knowledge of them now, the DNA by definition is double-stranded. So the same DNA that's in your cells and my cells are in bacteria cells. And the RNA is single-stranded. The reason I make that point, and I'm not going to really belabor it, when we get to viruses, you're going to see that viruses don't play by that rule. There's single-stranded DNA and double-stranded RNA in certain kinds of viruses. I don't focus on them. But because they're non-living things, they don't necessarily have the same set of rules that apply. All living things have a cell membrane. By definition, it's selectively permeable. It's a gatekeeper. Some stuff can get in, some stuff can't get in, and needs help to get in. So I'm not going to revisit diffusion, facilitated diffusion, osmosis, static transport. But effectively, that's kind of what living membranes are about. Phospholipid bilayer, embedded proteins, selectively permeable. Cell wall is not a criteria for a living thing. We don't have them. So in some instances, it's useful. You're going to see bacteria have them. We can be single or multicellular. We're multicellular. There's a lot of living things that are single cell bacteria. There's an example. We all respond to our environment. So if we're here, heavens, some of the rooms here, remember fall of last year, were particularly 90 degrees. You could hardly breathe in here. The windows wide open. No problem. We responded to the environment by whether it was opening the windows or, if necessary, not even having class in here because it's just too uncomfortable. We made a response. Bacteria, microorganisms respond, but typically they respond to, is there a certain chemical in the environment? Is there light? Is there air? Is there insufficient air, etc.? Those are the kinds of ways it responds to environment. Reproduction. See, you have to understand that bacteria don't reproduce like we do. When we talk about reproduction, we think of mitosis and meiosis. That's pretty much the way eukaryotic organisms like us reproduce, with some variation. When it comes to bacteria, very much different. You're going to see that they reproduce by something called binary fission. Very simple, very easy, very, very. So they do undergo reproduction because it's the only way a species can live. A living can continue to be a living thing as long as it reproduces. And then lastly, energy. That's like when I get to the bottom of the word, energy transformation. So that's a neat way for saying metabolism. The energy transformation. What do? We eat organic materials for the most part, along with other stuff, vitamins and minerals, and we break apart those organic materials, all that, those previous PowerPoint slides that I was showing you before, carbohydrates and proteins and fats, into their components. When we break them apart, break those bonds, they release energy. We gobble up the energy and use it for our own purposes, whatever they are. Make ATP, useful energy, take that energy and take some of the components we ingested and use them as building blocks for other things, for our own carbohydrates, proteins, fats, nucleic acids. That's what living things do. But this is about our concept today has changed. For instance, so when I was in school, we didn't even have the domain system. This is based on molecular biology. The concept of we had animal and plant kingdoms, and bacteria were something else, and we thought they were plants. So they had something. To this day, we talk about things that were fauna and flora, plants and animals. Okay, and we call them microflora. We talk about the normal bacteria that cover our skin. They really we call them, and you're going to run into people who still use the same nomenclature, normal flora, because it's a wall, even though they have nothing to do with plants. Just saying. So, I mean, there's a lot of misconceptions, but the domain system, and that should be WOESE, is the name of the principal researcher that was published on or about 1978. So I got my doctorate in 1977, sure, plus, and that we didn't have that. It's based on molecular biology, which was in its infancy in the late 60s and became more recognizable as we got farther along. And it's based on two things, this domain system. These researchers looked for similarities in ribosomal RNA, we're going to talk about that in a second, and DNA sequences, we'll talk about that. So you were able to chop up the RNA and the DNA and analyze it, and that's how he developed these classifications of the single-cell domains called prokaryote, because they didn't have a nucleus or other membrane-bound organelles. That's what made them prokaryote. Archaea, related to like archaeology, they only survive in extreme environments. The bacteria that are more or less everywhere. And what do they have in common? We'll get to why they're different shortly. They have a single cell and cell wall, all of them. But they're still living things. They've got their own DNA, different than ours. They've got their own RNA, by and large different than ours. They still do all those things. They reproduce, they have a membrane. In this case, they have a cell wall. They're not multi-cell, they're single cell. They respond to changes in the environment. Like I said, add a chemical, add water, add light, add air, take away water, take away light, take away air, whatever. They reproduce very simplistically, as you will see. And they do the same energy. And for the most part, the bacteria that you're studying in the lab do the same thing that we do. They're what we call organoheterotrophs, which means we basically eat everything, and we derive our energy and our carbon building blocks from what we eat. Organic molecule. That's what we are. So whether you're a vegetarian, a vegan, whether you're a pescatarian, whether you eat meat or you don't, or you eat fish or you don't, all of those things, you're still organic. You eat different kinds of organic molecules and get your building blocks from them and your energy. That's what they are. And we'll get into the distinctions between them. Interestingly, the microbial world doesn't have a whole lot, but they're very important. Karyotic cells. What makes them eukaryotic? Membrane-bound organelles. They may or may not be single or multicellular. They may have a cell wall or not, but they have got all the other stuff. So that's the jumping off point for this, my discussion of what we're going to cover that I have, because I'm such a bad handwriting, I've typed out for you. We actually just came from COVID. When we're, it was hard to have a board to write on. A lot of us got like little devices that you could sort of write on PowerPoints and save them and stuff like that. Dr. Hannah, who was just in here, one of my colleagues, she was the one who, clued me into what she was using a device like that. So the domains of archaea and bacteria, living things because they're made of cells, they're prokaryotic, single cells with a cell wall. Their DNA is different. Our DNA, everybody, is double-stranded DNA, just like bacteria's, just like archaea, except it's in pairs. Theirs is not. Archaeal and bacterial DNA is a great big circle of DNA that basically undergoes a phenomenon I talked about a little bit previously called supercoiling. You kind of fold it and twist it, fold it and twist it till it compacts. So even though a typical bacteria may be a millionth as long as we are, our body have between 35 and maybe 50,000 genes. genetic locations on our DNA strand. Bacteria have about 3 to 5,000. So even though there are a millionth, maybe 100,000 to a millionth as big as we might be, one of our cells might be, genetically they have about a tenth of the genetic material, so somewhat disproportionate. That's because of the supercoiling. That becomes important down the road. We squeeze in a ton of DNA in this little package by this coiling mechanism. So when a bacteria dies, effectively the DNA explodes out, and that becomes significant down the road. Just saying. So keep that in the back of your mind. That becomes kind of important when we get to our genetic unit. So single circular strand of the same DNA we have. A's, T's, G's, and C's, double-stranded. If the strand was one long strand, it would be like ours, anti-parallel. One side's right side up, an identical strand is upside down. Not a whole lot different than ours, except ours is a bit more advanced because we have pairs. And the pairs help particularly with regard to inherent genetic stability. The term carry-on, if you recall, maybe in high school, when they showed you Down syndrome, trisomy 21, they showed you what we call karyotyping. We didn't have the ability to genetically look at the sequences like we do now, but you could really you could extract the DNA actually and look at one of our cells and look at the way the DNA looked. That was called a karyotype. And you'd see, here's the first, the second, the third, and we get to the 21st chromosome, and voila, you would see 3 of them rather than 2. By something that many of you study called non-disjunction. They just didn't separate when they were supposed to during meiosis. Okay. In this way, and a lot of times we'll see when you see defects or irregularities in chromosomes, it usually has significant implications. Fortunately, here we're adding one rather than taking one away. When you take one away or take a part away, it generally has more significant complications. Down syndrome folks live long, productive lives, and we underestimate them, even though there is a certain amount of mental compromise. Okay, that's associated. They're clearly mentally disabled. We used to use a word beginning with an R that we're not supposed to use. That's now become a pejorative term. So I'm not saying it. Someone will say, he said that word. Just saying. See, I see somebody, they got their phone out, they're ready. Even though I'm recording this for posterity. So I'm trying to be politically correct, at least occasionally. So what do they have? What do we know about them? They don't have, what makes it a nucleus different than that super folded, compacted? It's got a membrane around it. And that membrane is just like a cell membrane. It allows traffic control membrane. You can come in, you can go out. It's like you want to go onto the plane in the airport, don't have a gun in your pocket. They're going to get upset at the TSA. They just are. Right? You know that. I always set off the metal detector. Just because I have a steel suture inside of me. And I've always got the wand. And my wife's always giving me a hard time. It's him. Okay, so basically that's what the nuclear membrane is. Membrane-bound organelles are a degree of sophistication. It means we can do things in membrane-bound compartments, and activities that go on in there are sort of sheltered from what's going on elsewhere in the cell or other membrane compartments, like a mitochondria or a lysosome or a peroxisome. It's a good thing. Okay, so I mean that's really what membranes do. Is they do allow traffic, but we control the traffic. And that makes it a much more sophisticated. So our concept of a bacteria is kind of like when you used to be in Pittsburgh and when you go to the grocery store and you get a plastic bag. And you have like 8 things and you throw them all in. Oh, that's a bacteria. No infrastructure. It's like no compartments, no nothing. It's just like we got a plastic bag. That's the membrane. And here are all the organelles that are in there. That's it. And we realize now that that's not true. With the advent of sophisticated microscopy, and sophisticated techniques in molecular identification, we now know that there's an infrastructure that's there. Not nearly as advanced as when you guys are in AP1, all the way the cells were and how you had motor proteins and they move things all over the place. You might have come in and just saw I was doing a muscle physiology unit with my advanced physiology, all that stuff with the sliding filaments that you learned about. Okay, that they haven't quote unquote had yet in this form that I'm presenting it to. But that's a degree of sophistication. Now, the big thing that the domain people in their research looked at were differences in ribosomal RNA, bacteria, and it's called an S unit. You don't have to know what it means. It's named for a physician or a scientist named Svedberg, Svedberg. We just call them S units. You don't have to know Svedberg's name. And what happens is you have a small and a large subunit, and when they come together to make a ribosome, the density actually gets lower. It's like a 30 and a 50, which a total would normally be 80, but when they compact together, the density kind of decreases to 70. So it's a very interesting phenomenon, where in our eukaryotic cells, the vast majority of the ribosomes are 80S. They're larger and they're, if you will, more densely associated based on these units. They don't have the membrane-bound organelles. We have a nucleoid, not a nucleus. They don't have anything to do, almost all of them replicate by binary fission. And that's the secret sauce why bacteria is such a pain in the neck. The energy they produce is basically dedicated to one thing, reproduction. And so every 20 to 30 minutes, they go from 1 to 2, from 2 to 4, from 4 to 8, to 8 to 16. It's called exponential growth. We'll see it graphically when we get to a subsequent chapter, when we talk about growth and the factors that influence it. And that the big problem with that is in a day, if you take E. coli, okay, which you work with in the lab constantly, and maybe you played it out in your lab this week or will be, okay, and you're going to watch it grow. In a matter of, you won't see anything on the plate now, but if you came back in tonight and certainly tomorrow and looked at those plates you stuck in the incubator, you see growth right away. In order to see bacterial growth, we call it a colony, you have to see, are you ready? At least 1 million bacteria in about 21 hours. With binary fission, you get about 1 million bacteria from one. So if you started with 10, you have 10 million bacteria in there already. Pretty good. That's why it's so important to treat infections early. Because if you don't get a handle on them in the first 24, 36, 48 hours, with a diabetic with a bad infection, they could die easily. Immunocompromised person with a bad infection, they can die. So that's part of sort of the secret about them with that. So they replicate by binary fission. Again, put something in the environment, they'll respond. And again, most of their energy is derived from organic compounds, just like we do, building blocks and energy. Bacteria, there's something unique. It's a magical material that's in there. Nearly every bacteria has it in their cell walls. It's not always enough to be of significance, but it's there. And it's the single feature that you're going to do with the single most important stain you do in your bacterial lab called a Gram stain, named after a Danish physician. It's a proper name, GRAM. Okay, so it's not a measure of weight like, milligrams or kilograms or anything like that. And it's called peptidoglycan because it's a mixture of peptide bonds, amino acids chained together, and glycans, which are a chain of sugars, except the sugars have nitrogen. That's why they have the N at the end. It's not glyco, it's glycan. So nitrogenated carbohydrates, which we use a lot for structure in our bodies too. A lot of connective tissue is made. A lot of that, remember the matrix in connective tissue and the ground substance you learned about in AP1? A lot of that is nitrogenated carbohydrates. And this one has embedded amino acids. And when you say unique doesn't just mean unusual when it comes to science or a biology class. Unique doesn't mean important. Unique means that only bacteria have it. Nobody else does. That's what's unique. Unique's a big word. Okay? We don't have peptidoglycan in our cells. Bacteria do. So when you have something like that that's unique to bacteria that has nothing to do with us, and since it's an integral component of their cell wall, if you destroy the peptidoglycan or moreover stop it from being able to be made during this massive reproductive cycle, You can kill a bacteria. That's right. You, ladies and gentlemen, can kill a bacteria. Just call this number. We just need your credit card. And yes, you can kill the bacteria. I sound like I'm on one of those infomercials. Like QVC. The Home Shopping Network. You too can kill the bacteria. As long as you can attack peptidoglycan. So when you have a staph infection or a strep throat and you go to the doctor or you go to urgent care or you go to the ER, what do they give you? Right, they give you, and what is amoxicillin is what? It's an antibiotic. Yes, you both have it right. Amoxicillin is a kind of penicillin. It's a chemical cousin of penicillin. Penicillin, if you did not know it, many of you are growing in your refrigerators in your dorms or at home. It's that kind of blue-green mold on the white bread. That's penicillium species. So just have some moldy white bread. It'll fix you right up. Penicillium is a very, very common fungi. And so what it does in penicillin, when you refine it, it has a chemical structure that interferes with peptidoglycan being assembled. Peptidoglycan is a polymer. So it's a polyprotein. So if you can't put the pieces together and it interferes with their production, you effectively destroy the cell wall. And if you destroy the cell wall, you destroy the bacteria. That's the good news. It is what we call it's a fancy word we're going to talk about in antibiotics called selective toxicity. It's going to kill the bacteria and it really will have no effect on us. What's the big problem with penicillin? It comes from a mold. The big problem is allergies. The three saddest words a doctor or a nurse can hear when you do a physical are this: what are you allergic to? I'm allergic to penicillin. Because it just took away penicillin, amoxicillin, methicillin, most cephalosporins like Keflex, which is cephalexin. More than half of all antibiotic prescriptions are that family. If you're allergic to it, you really have to be very careful. If you're going to use it, you have to test first to make sure. We talk about that in our antibiotic unit. So that's really important. Now, on the other hand, if someone is, and people are allergic to penicillin, more people think they're allergic to than really are, but that's a whole other thing we go into in antibiotics. You have to find something else. Well, the big difference between archaea and bacteria is the 70S ribosome versus our eukaryotic cells, which have 80S ribosomes almost entirely, and that's the sticking point. So if someone's allergic to penicillin, and maybe someone in class is, what do they give you for an antibiotic? Other than penicillin or amoxicillin or ampicillin or augmentin, all of which are penicillin-containing things. Has you ever gotten a different kind of antibiotic, one that worked fast, one you only had to take a few doses of? There's the word Z-Pak. Aha, I'm seeing recognition. Did anyone ever get a Z-Pak prescribed to them? Okay, Z-Pak stands for azithromycin. It's in the family of what are called the drug of choice for penicillin allergic individuals. Drug of choice in addictive disease is the drug that the person who is addicted, whether it's alcohol or heroin or cocaine, would they prefer to use to get high. In medicine, it has nothing to do with. Drug of choice is the drug you're supposed to use for a particular diagnosis, unless you have a very good reason. You know, what's the drug of choice for this? It's penicillin. Why didn't you prescribe penicillin, doctor? My patient was allergic. Oh, okay. Next question. You don't want to give somebody an opportunity. You have to have a good reason. They can't use it. They couldn't tolerate it before. It made them deathly ill before. Fine. Now I've got a good reason why you didn't use it. So it's from the family of erythromycins. Erythromycins target the other difference, the ribosome. Now, the good news is they target the ribosome. And the two largest categories of antibiotics are ones that attack the peptidoglycan-based cell wall or the 70S ribosome. The vast majority of antibiotics are in those two categories. Unfortunately, we inside of our mitochondria, and all our cells basically have mitochondria, have 70S ribosomes. So the big problem with using, say, the Z-Pak for everything is side effects. And side effects and allergies are not the same. Side effects mostly are gastrointestinal. Constipation, diarrhea, upset stomachs, vomiting, all of those things many people have gotten. And for you going into nursing, you're going to be sick and tired of hearing about that because you hear it all the time. Or from there. That's why this basic information is so important. That's why in the next chapter we do, when we really look at in depth the anatomy of bacteria, why we focus for a day or two on gram-negative versus gram-positive. Because that's where the peptidoglycan is, and it's the peptido being able to attack it really determines not only what antibiotic, but different ways of growing them and dealing with them, and a whole subset of problems. You see, what bacteria do to hurt us is those waste products. They don't. It's not like they're magically going to damage a lot of cells. Sometimes they do, sometimes they don't. Most bacteria produce toxins because they're different from us. Yes, ma'am.
Like, what about the bacteria?
That's the biggest problem we have today. We have to either find an agent that we can use successfully without hurting the patient. If not, we have to find some other means. And that's the difficulty. You're going to hear a lot about superbugs, bacteria, where we're running out of alternatives. And we're actually looking at viruses and other treatments to try to deal with that. Fortunately, right now in this country, we haven't encountered ones. The trouble is sometimes those antibiotics are so severe, and you have to be very judicious. It's a very good, very perceptive question. Very good. That's the big problem. And that's part of what I go on about a lot in this class, is why we've gotten to this point and what we can do not to be at this point. Because now we've recognized the problem. We've overused and inappropriately used antibiotics. When I was a kid, everybody, you have a cold, go take some penicillin. It's a virus. It doesn't work anyway. You're treating the parents. For you in nursing, you're going to run into this. Why didn't you give them any medicine? You just told me to go get Tylenol, or go get ibuprofen, or go get, Zyrtec or something. Hey, because that's what you need. It's very hard. Parents want you to, I want a magic pill that's going to make little Johnny or little Judy better. Moving on. So they live everywhere. Okay. Archaea, extreme environments. What's interesting is the distinction between them is based on different sequences of ribosomal RNA, and because we can chop up DNA, interestingly, archaea, the ones that live in these extreme environments, polar ice caps, bottom of the ocean, volcanic caldera, have more similar sequences to our DNA. And scientists they're not making a big deal out of it. But it's kind of like, if you think when life began, the planet was a very inhospitable place with extreme conditions. And maybe the earliest DNA is still part of us in some ways. That's the only sense I can make of it. They have cell walls. They have something that looks like peptidoglycan, and they have some different things in the cell membrane that can withstand the extreme environment. That's the big difference. And this is about the only time we'll talk about archaea, other than like as a test question. Well, how do they differ? They're the same size. They have a cell wall. The environments that they live in, maybe some aspects of their DNA sequences, and that's about it. So we'll get a good look at this, which brings us to the other half of the family, eukaryotic pathogens. So to be fair, most of this course is about bacteria and viruses, but they're not the only players. Eukarya, the big problem with them is the cells are like our cells. So if we try to give us something to fight one of those infections, typically they have a lot of side effects because they're like our cells. Okay, and a lot of these things live in our cells, so we have to damage our cells to get to them. There's big problems with that. So animal and plant cells, but they're macroscopic, single or multicellular. Most of the ones we're dealing with from this course are single-celled. They don't have to have a cell wall. Some do, some don't. It's not a big deal. Okay, they have the same kind of DNA we do, and different pairs of DNA, double-stranded, just different numbers in different organisms. Again, the aforementioned 80S ribosomes. So that's a big difference. And typically, there's other forms of reproduction. Most of the time, mitosis, and it should be MEI, I misspelled it. When you see yeast do something called budding, you typically see that in the lab. And when you look at fungi and mold, you generally look at prepared slides, and we classify them based on how they spore. So they release spores, and that grows more of them. There's different ways to look at it. It's not terribly essential because most of you don't do it. Well, I dealt with a lot of skin fungal infections, so we used to take skin scrapings, and I'd look them in my office under the microscope. You'd stain them and look at them. So I mean, it depends on your area of what you're working with. If you're working in a dermatology clinic, you end up doing a lot. So understand that those are some differences that are there. The ribosomes, the density and the pairs of that are there, they live all over the place, but the highlight film is they have a membrane-bound organelle system, including a nucleus. And we're going to go over all this. The major divisions. The first one is very interesting, and because that, after bacteria and viruses, yeast and fungi are probably as important to us as anything else. Okay. I mean, vaginal yeast infections are a big-time problem for childbearing women, immense. And they're brought on by strong antibiotics. I've treated so many diabetics over the years that are part of that. And they're also when yeast typically are two forms. There's a little yeast you see under the microscope that you might use a little packet of for a recipe for cooking or baking. Okay. And they're ones that form strands, and they get like a tuft, and they're called mold. And mold's a big problem. Again, because it can be in the air, it can harm people. There's diseases associated with it. So they have cell walls, so they're like a plant, but they're not because they don't have any chlorophyll. They are land-based, not water-based. And the way they do business, the way they gobble up stuff is they decompose it. They secrete enzymes. We eat something, we make enzymes in our stomach, in our mouth, in our small intestine, and we absorb the nutrients. So it's not necessarily within the context of the organism. When they grow, when fungi or yeast grow on something, they secrete enzymes that break it down, and they do it for dead and dying, decaying stuff. So they're this way called decomposers or saprobes. The fancy name is exodigesters, which you don't have to know. Okay. So when you see roadkill decomposing that hasn't been cleaned up, some bacteria do it and some fungi do it that are there. Algae, talked about before, they're the only microorganisms that really have chlorophyll. That distinguishes them. We have pigmented bacteria that behave like that. They don't really impact us that much. But algae, which are water-based, have a cell wall, can be single or multicellular, and we talked about the importance of them producing oxygen on a planet-wide basis. They're in the subfamily of protists. Okay. And the other member of that, and I didn't write the name down, or I did rather, are protozoans. Protozoans, okay, not protists, but the protozoans, I should say, have a single cell, no cell wall, no chlorophyll. They can live in the soil or live in the water. And they are really significant to us. I talked about malaria. That is an organism that exists in the bloodstream and flourishes in the bloodstream of certain kinds of insects, particularly certain kinds of mosquitoes. And that's what yields the dreaded disease malaria. Again, still the largest killer in equatorial Africa. Affects untold billions of people, large amounts of people. Not AIDS. That the second most common sexually transmitted disease in this country by number of new cases. First, most authors will tell you is HPV, human papillomavirus. Third is genital herpes. In the middle is something called Trichomonas vaginalis. That is a protozoan disorder. So understand, it's not like they're innocent. They're there. And if you know somebody with contaminated well water producing really sick people with diarrhea and GI disorders, typically they're protozoans like amoebic dysentery or giardiasis. So they're in that as well. We're not done yet. Because this is just the cellular guys. We have acellular forms, by definition, non-living. And so, and I'll fall into the trap. Yeah, all you have to do is walk and look at some type of antiseptic or disinfectant, and it'll say rapidly virucidal. That means it kills viruses. You can't kill them; they're not alive. So it's already, we use the term because, oh, does it kill viruses? Yeah, sure it does. Because you and I, the individual, the layperson, understands they just want to get rid of them. The reality is it can render them inert if they're active, or destroy the virus particle, depending on what type of agent it is. And we go over all the agents. So by definition, they're non-living because they're not made out of cells. At the very least, and the trouble is they live inside of, or damage, or both, living cells. Most of them are obligate intracellular parasites. In order for them to live, they invade a cell, a living cell, and they can invade any living cell. Bacteria can get a virus, you can get a virus, archaea can get a virus. They invade any or all of those cells, even though there are different types of viruses that do that. And so effectively they take over and hijack the machinery inside the cell. That's how they exist, and moreover how they reproduce in a unique fashion that's nothing like binary fission, mitosis, or meiosis. All we know about them, really, and we know a lot comparatively, is that they have either DNA or RNA, never and a protein shell at a minimum. The common cold virus would be just like that. Okay. When I first learned about viruses in my micro course in 1974, I wasn't taught by somebody who had an idea what viruses are. This was the lead virologist of the Commonwealth of Pennsylvania. The one who did our virology. He went up and drew something that looked like a bullet, and did that, and said, this is what we kind of know viruses are back then. We know it's got a protein shell, and it's got some type of nucleic acid, RNA or DNA, period. That's about what we knew before the age of molecular biology and advances in microscopy. And you sort of went from there. Yeah, they go into cells, and yeah, they damage. Most of the damage isn't by making toxins. Most of the damage comes from altering cells and the immunological response, our immune system going after them. And when you talk about a flu or a cold, that's how viruses do most of their damage. Bacteria is mostly toxins, though, to be fair, some of them cause immunological harm. COVID, big time, it's all about your immune system responding to it. And in some people, the healthier they were, the worse it was. Because a healthy immune system means a very overly vigorous response. Sometimes these new or novel viruses, what makes them really, really tragic is it's not the very old or the very young who we typically associate with susceptibility. It's the healthier individuals. That was true of the Spanish flu, which is a novel virus, which turned out to be a bird flu because there was bird genetic material in it. That was the largest single infectious disease killer proportionately ever. I mean, so yeah, there's plenty of historical evidence about that. So now, to be fair, they may also have a membrane. It's a membrane, it's a phospholipid bilayer. It's different because it's really not the semi-permeable membrane. It's just an additional enclosure. And it's interesting. COVID, for instance, is an enveloped virus, whereas what I was taught, rabies, which I drew up there, is also an enveloped virus. They just didn't know about the membrane. HPV, or the common cold, we refer to them as naked viruses, not for anything having to do with clothing, but the absence of a membrane. So non-membranous or non-membrane enclosed might also be used. And again, all living cells can be affected. And lastly, in our list of microbes that are bad are these misfolded proteins called prions, which is kind of taken from protein-based infectious particles. So they're only proteins, they're misfolded, and they bump into other proteins once they're in your system, mostly in the nervous system, and cause them to misfold or tangle or become irregular and non-functional. And we'll get more, because we do a whole chapter on viruses, and the prions are at the end of it. And the one that most people at least have heard about, is mad cow disease. And so that's all that handout. Now, as we go back, as we get ready to finish up in the last roughly 8 to 10 minutes today, I go back to our original PowerPoint, and just to finish up with a few random thoughts. Same thing, single cell, bacteria, protozoans, animals and plants much larger. When we get into classification, we'll look at the different shapes because that has a lot to do with how we identify them, particularly microscopic shapes, how they group together, staining patterns, chromosomes, ribosomes, complex functions. These are all different kinds of cells. Here are the eukarya again. And see, here's another error on the PowerPoint. It says it does have other organelles because understand that bacteria have DNA and RNA. They're living things. The ribosomes are in there, just not membrane bound. They're not those bound ribosomes you learned about in AP1. They have ribosomes that make proteins. They do. Absolutely. And yes, now we know they have an internal structure. And yes, they can engage in the same activities. And they reproduce and they respond to their environment. And they undergo energy transformation. It's just all that stuff I wrote about that's living. Okay? Which brings us to the last part, which is taxonomy or nomenclature. And I'm sure that in high school biology, you went on family, classes, orders, genus, and species. Is it ringing the bell? Vertebrates and invertebrates. Yes? You heard about that stuff? You know, like my personal favorite, you know, arthropoda, which are insects, insecta, because you have like shrimp in there and lobster in there. Yes. Or, you know, hey, crustaceans. So, but we don't have to do that. We don't have to do scientific names and classify and go bananas with all that stuff. We do it the easy way, genus and species here in micro. The genus, by definition, is capitalized. And so this is E. coli. And it was named, because scientists are vain, okay, after a guy named Escherich, who discovered it in the colon. So the second word is its location, and the first word, the genus, he took out of his name. And so who wants to keep writing Escherichia? So we just write E, period, coli. And that's perfectly legal on a chart. If you want to write S. viridans, okay, or S. pneumonia, that's perfectly fine. Okay. The only thing I would recommend you do is to underline it because it's a little hard to write in italics. So we don't do this. We're just interested in the genus and the species. Binomial nomenclature, two names. Like we're Homo sapiens. Everybody knows that. Capital H for Homo and sapiens is all in small letters. And they do that. And again, here, this was the idea. When you start talking about that particular pandemic had evolved and being able to infect non-human animals, and then so effectively it's, and you're going to see that with influenza and you see it with COVID, that multiple species can get that, and they have a unique way, sort of a special way of swapping genetic information. And so all of a sudden you get a combination where you'll have different kinds of DNA. If you saw, and it wasn't a great movie, if you saw Outbreak, the one with Matt Damon, Contagion. That was the one with Matt Damon, okay, and Gwyneth Paltrow, and that Kate Winslet was in that. That's not a great movie, but it's very much what they do, and that was the investigation. They had to backtrack from her being patient zero, you know, what we call the index patient, and backtrack to where she contracted it from, and basically from some type of tainted food that was on a farm, or she was on a farm where the food was, whatever. Movies, but that's relatively accurate from that. So there's a lot of good information that's there. And so these are the other models that were used. We used to, when I was animal and plants, and something called Monera, which was really where the bacteria came from. And then I graduated more to the Whitaker model, which is what we use. And this is what we're talking about, these small ribosomal subunits that Weiss-Fox, and Weiss was typically credited, and proposed these three domains. And so it's something you should be familiar with. And that's everything for today. So we're going to start with, I think it's chapter 3, which is really, really looking in depth in the bacterial anatomy, the nuances of it, and specifically gram-positive versus gram-negative. Yes? Beg your pardon?
The notes.
Oh, you want to see the notes?
I just missed it.
This? Great. I'll see everybody Friday. I know I'll be here. I gotta be here anyway. Sorry, no virtual. I'm saving them. You gotta do 4 lectures every day, and you're stuck being one.
We have half from 3 to 6.
When?
Right after that. Right now.
Oh gosh, really? Who's doing your patho?
Margaret Rizzo.
I don't know. Thank you.
Thank you. You know what you're from?
Oh,
I didn't need that.
I'm sorry. I remember when they had Dr. Pio do it. Oh my God.
I think he still does it. It's just
She,
or
Katrina.
she, sorry, yeah.
Yeah.
I am. We just there's only one section.
Oh, really?
Yep.
Yeah, patho's hard.
Question.
Yes, sir.
Most of your exams, do you base them off the slides, or you're mostly what you give out on
But
your?
a lot of it is on the printed notes that I have, and I'll do a review recording. So, and really, my points of emphasis in class, like today.
Yeah, that's
you
what I
already
was-
know, algae are the only ones with chlorophyll.
Yes.
The acellular ones which are not made of cells, are viruses.
Mhm.
And how do they differ? Well, the protein,
Protein,
prions
yeah.
are protein only. Viruses, DNA or RNA, never both. Protein shell, maybe membrane. Those are like 2 or 3 test questions right there.
Yeah,
So
that's what-
points of emphasis would be the way I would.
Thank you. 'Cause,
I would,
yeah, I was
would-
making.
would
'cause I was
play it
writing-
out for you.
I was taking your notes and then writing over with mine, and I was making sure